We have a lot of factors that are influencing healthcare today. We have people living longer with chronic disease. We have emerging market opportunities around the world. We have new models for healthcare delivery, and we have the rise of the consumer patient influencing the choices that are made in that healthcare. As Dr. Califf mentioned earlier this morning, we have the promise of personalized medicine and the big data challenge that comes with it. And in addition to these factors that are shaping healthcare, we have opportunities that are giving rise to new medical products. Device technology is accelerating, information technology is making more possible, and we have the marriage of biologic with those two other factors that are creating opportunities for new products at an accelerated pace that we've never had in the past. And along with that, our patients, our payers, our regulators, our caregivers are all increasingly asking for more evidence, pressuring more evidence development, more knowledge of how products perform, and that's impacting the clinical research costs. So this is a picture depicting that increase in cost associated with developing clinical evidence. And we believe that models and existing information and leveraging those models in existing information are a way that we can drive down this cost curve in the future. So at MDIC, nearly exactly 2 years ago, we kicked off an activity where we were going to take computer models and we were going to combine them with existing evidence to inform clinical trials to reduce the amount of clinical trial data that was necessary in a prospective clinical trial. And the idea was in the future, we would start to leverage more computer models, and we would introduce this concept of a virtual patient. We are building on the FDA's guidance document on the use of Bayesian statistics that gave an opening for using informative Bayesian priors as part of the clinical trial design. And we believe that we could use computer models and existing evidence as that informative prior. So this group at MDIC set out to build a statistical framework, a Bayesian statistical framework that would incorporate this prior information, but guard against bad information by having mechanisms where that information would be discounted out of the clinical trial. And with this new model, we would be able to maintain clinical endpoints, do the same work that we already do, but potentially with reduced clinical patients in the new clinical trials. So once we created this framework, we approached our partners at FDA, specifically the Division of Cardiovascular Devices, and we asked if we could conduct a mock submission. We were inspired by work that had been done within the Office of In Vitro Diagnostics in 2010, where they had conducted a mock submission. They'd had great successes, and they'd learned quite a lot about what a mock submission can do and how to operate one. And we made a proposal to Bram Zuckerman, and he kindly agreed that he thought investing time and effort in conducting a mock submission would be mutually beneficial. It gave us a shared learning mechanism where we could envision innovative regulatory science, operationalize it in a collaborative approach, and to do this, we set up a sponsor team that represented MDIC members from across industry, service providers, nonprofits, and FDA members. And then, through their normal process where they identify a lead reviewer, the FDA created a review team that was distinct from the sponsor team so that this review would occur just like a typical review occurs. And the sponsor team had knowledge of the mock submission only through our formal interactions during our meetings and our written notes and questions that we received from the review team. In a spirit of openness and transparency, all of the work that we created is on our MDIC website. The three Q-Sub meetings that we conducted and all of the presentations that were given are available, so you can do a deeper dive. Today, I'll give some highlights of that mock submission and some learnings that we had. So we started this in 2014. Just under 2 years later, we've completed that mock submission. We have a planned workshop, the first of which will occur at AdvaMed next month, followed by a larger workshop after the first of the year. So for this mock submission that we conducted, our example was lead fracture. Lead fracture prediction served as our computer model that we were able to use as a Bayesian prior. And the endpoint in that clinical trial is survival from fracture, or the length of time that the lead is operable, making sure that it extends beyond a certain minimum time. And because we're talking about virtual patients, we have to represent the true variability that you see in the population that receives these medical devices. This includes how the device is implanted, the bending conditions throughout normal use cases of daily life, and how that impacts the motion of that lead, as well as the material properties of the engineered lead itself. And all of these serve as inputs into the computer model that was used as that Bayesian prior, which allowed us to simulate the entire family of possible outcomes that could occur, the survival curves for these leads, which you see on the right-hand side under the outputs. It allows us to virtually propagate the variability and the uncertainty that we see, both the human variability and the engineering variability. And this particular graph is available in a journal publication should you like to read further details. So the concept of how you incorporate these informations, whether that's a computer model or other evidence, into the clinical study through this virtual patient concept is that you need to have a model that is an accurate representation of the patients that you're going to enroll in the clinical trial. And if that model is mature, you can discuss how many virtual patients are available to include into that model. You can talk about the statistical rigor that has to exist to make sure that you conduct a clinical trial with controlled power, type 1 error, and confidence intervals. And then through the statistical framework that MDIC created, the virtual patients are allowed into that clinical trial as prior information when they're concordant with what's observed in the actual running of the clinical trial, and thereby potentially reducing the number of real patients that are subjected to the rigors of the clinical trial and that investigational device. When the clinical trial data and the virtual patient are discordant, the virtual patients are decreased and excluded from the clinical trial. Conceptually, that's the framework. For all of these statistics and the how-tos, there's a journal publication that we all know, the time constant of journal publications. We've been working for about 18 months now, and it is in final review. We've finished our responses to comments, and we're expecting a positive outcome and publication notification in the month of October, November of this year. And it's the incorporation of stochastic engineering models as prior information in Bayesian medical device trials. As you can see, the associated working group that helped produce this document and helped produce this mock submission are listed here. We had excellent collaboration from the entire MDIC ecosystem, and we believe that this is an opportunity to create a framework that uses all the evidence that's available to help accelerate access for patients to novel technologies, to new technologies in a safe and timely fashion. So with that, I will hand it back over to Dan to introduce our next speaker.
Great. Thanks, Don. In addition to the mock submission, one of the projects that we've been undertaking in ACTP has been applying the statistical model to some retrospective trials. So that was utilized as a low-risk way to build some learnings around the model. So to discuss that project more, we have Roseanne White. Roseanne is the Director of Pragmatic Clinical Trial Statistics with Duke Clinical Research Institute. Roseanne?
Good afternoon, everybody. I want to thank MDIC for allowing me to speak with you today about a topic that I feel very passionately about. In looking at the MDIC alternative evidence-based design, we're looking at a high-level view of the virtual patient project. We have the foundation, which is the FDA guidance document, Our focus is looking at the simulation of the clinical trial processes, our outcomes using existing clinical data, and the challenge is to determine if the model can optimize, decrease clinical trial enrollment requirements. So, how are we actually doing this kind of work? And I am going to sort of make it grounded a little bit for you. Suppose you have historical data, and I'm going to represent this as cans of paint, of course in the Duke blue, because I am at Duke. And we have historical data. We're going to paint a room. We've got some old paint. We're going to buy some new paint. We're going to mix them together to paint the room. However, if the current paint that we get is the same color, we're going to mix all of it together so that we will have the right color. If the current paint is a little bit lighter, we wouldn't necessarily mix all of the old paint with the new paint because it would really change the color of the current paint. And if it's really different, then we only use a little bit of the old paint in with the current paint so we maintain the color. And so that's what we're doing when we're using the historical data. We have the historical data and the current data. We compare the two, and we use enough of the historical data such that we maintain the integrity of the current data. So the way the process works is we look at the historical trials or model output whose population and treatment measures are similar to the new trial. We compare the new trial outcome results to the historical trial outcomes. The closer the new trial outcomes, the closer the color of the old paint is to the new paint, the more information it contributes. And in the Bayesian, we weight the historical data with the current, and the weights are based on a loss function. So what do I mean by a loss function? Well, when I was showing you in the different amounts of paint that was being poured into, we have to have some sort of independent measure of how much paint we're actually going to add to the current paint. And so what we do is develop a loss function. And this is where I think what makes Bayesian, I think, a little more unique than some of the other statistical methods is because this needs to be a collaborative method, collaborative effort between the statistician and the clinical scientist. Because in order to determine what this kind of loss function looks like, you need to be able to control type 1 error, which I refer to as false hope. Type 2 error, missed opportunity. In other words, you have something and you missed it. And bias. In other words, what is the smallest meaningful shift in outcomes that you can allow and still feel that you're maintaining the integrity of the current data? Again, going back to the paint example, how much can I allow a little bit of difference in that paint? And when I paint the wall, nobody will really tell the difference. And so to be able to do that, you need to be able to talk with the clinical scientist. The clinical scientist needs to be able to tell you, well, I think this is a clinically not meaningful shift. This is the kind of type 1 error and type 2 error I'm really looking for. And the two of you work together to create this loss function. And depending on what the outcome looks at, you go then to the loss function to figure out how much of the historical data that you use. In our case, what we did is this with a retrospective analysis. We looked for situations in which industry had a current trial that was being done in the United States and a historical trial that may have been done outside of the United States. And so we used the earlier trial as our historical power prior, and the current trial that they had completed in the United States is the current. And what we did is to try and see if they had done a Bayesian power prior, how many patients would they have saved and still maintain the quality and integrity of the result of the current trial. And we went and then collaborated with each of the different sponsors. They did their own analyses. What we did, and what my role is with MDIC, is to help consult with the individual industries so to help them understand how to apply the applications. but each industry member did their own analysis. And we feel this is a very important part of this program because it's part of the education. We do not want to come out with a technique where each individual industry cannot figure out and do it for themselves. So we want to make sure that we are enabling everybody, empowering everybody to use these techniques. And then they do a closeout where they report the results, and then we'll be putting out that information. Let me tell you where we are right at the moment. And the types of savings that we're seeing. In a diagnostic trial where we have a single arm versus an OPC, the enrollment total was 150. We were able to use 75 virtual patients, which meant a study savings of 50%. In a vascular, which was a test and control, where we had information for both the test arm and the control arm, we were able to we used 21 virtual patients and saved 16%. In an orthopedic, single arm versus an OPC, we saved 32 patients. And in an orthopedic single arm versus historical, we were able to save 121 patients. So it ranges from 16 to up to 50% that can be made in savings. Once again, a lot has to do with how close, in terms of outcomes, the historical data is to the current data. But there is the saying that people who do not learn from history are doomed to repeat it. So in this case, what we're trying to do is not repeat history, but augment history. And so by using these particular methods, we're no longer doomed to necessarily repeat it. We're hoping to go into the next phase, which is a prospective process. This case, we're going to go with an industry partner who is currently designing a trial. They do have the historical data, and we're going to actually prospectively do this and then see how it works in terms of how much savings we actually do get. In my particular industry, where I was working for the last 25 years in biotech at Guidant, we had already been using Bayesian clinical trials because we were doing metallic stents. And for those of you who are familiar with that, we would do generation after generation after generation of stent. And so we had been submitting these kinds of proposals all the way along. So I feel quite confident that we're going to be able to see, in this case, the types of benefits that we saw back in the metallic stent days in terms of using the Bayesian power prior. Thank you all very much.
So, thanks, Roseanne. Turning back to the panel, what we'll do just for a format is we've got about 4 stock questions, so we'll go through those questions, and then as time permits, we will open questions to the floor, or I think we maybe have the opportunity to take some from the internet or the WebEx as well. So, given that, first question is going to be for Owen. Owen, given the recent FDA guidance on adaptive designs and real-world evidence, what are the best ways to engage with FDA in discussions of innovative clinical study design efforts, such as incorporating MDIC's data aggregation model?
Sure. Thanks a lot, Dan. Good afternoon, everybody. I see both of these guidances as being part of a continuum of CDRH's effort to increase the collaborative nature of our interactions with external stakeholders, to improve our flexibility on different types of data that we might see for different regulatory submissions. And I think it really all ties back to one of Jeff's first slides this morning about our goal being to bring safe and effective medical devices to U.S. patients as quickly as we possibly can. And to do that, we need high-quality data. And there are times when that high-quality data needs to be the traditional randomized controlled trial that enrolls a lot of patients and follows them for a long time. There are times when that's the right thing to do. But I think we all recognize that the way of the future in many device spaces is for us to be more nimble and more flexible, to see less expensive and more and faster clinical trials. And frankly, there are some spaces where the way of the future is that we don't actually need a clinical trial to answer certain questions. And to take that even further, I think there are many device spaces where the future is in fact now. And so I think these guidances here are really intended to start to articulate some of those concepts where, frankly, the message is we are open for business. We are open to have these discussions with you about different ways that we can accept data that will support your regulatory submissions. And there are many spaces where we and industry are very comfortable with, say, Bayesian designs in certain device areas and less comfortable in other areas. And I think the project with MDIC of taking this into a little bit further with the Bayesian model on the virtual patient concept, I think really it lays it out in a very understandable way for folks who may not be intimately familiar with Bayesian statistics, and I think that's really helpful. And I think, frankly, it will be helpful for the dialogue with FDA as well. Not all of us are Bayesian statisticians on the FDA review teams. So I think, that, as well as the real-world evidence guidance, really just lay out that we understand that there are different ways to make your case and to demonstrate with high-quality data that a device should be brought to the market. There are ways that we can use these data for other questions about how our device is performing afterward. There are very low-lying fruit that we now see much more commonly where we approve a PMA, and maybe a few years ago we would have said you now have to do a standalone post-market study that's going to last a long time, it's going to take a lot of patients, and frankly it's going to cost a lot of money. And we now have PMAs where we approve, and instead of saying you need to do this standalone study, we say you need to take part in this existing registry. That's a huge step forward. We can go much further than that, but that's a huge step forward even in and of itself. And so these guidances really lay out how we think about these kinds of data, how we assess whether they're reliable, whether they're robust, whether we can count on them, and the kinds of questions you might expect to be answering. So now to get to your question, how do you engage? I'm just going to give a little bit of a plug for our pre-submission program. That's really where you start. It's not helpful to come in with a clinical trial application or a marketing application with something that's this novel and have that be the first time we've had a discussion with you. Coming in earlier, having that discussion is really helpful. And I do want to emphasize, when things are new like this, I can't tell you that the first discussion you'll have every time will be a positive, collaborative, yes, we're ready to hit the ground running with this new, exciting, innovative idea. We have to get up to speed as well. And so coming in as early as you can, bringing these ideas, as much justification as you can, and recognizing that if you've been intimately familiar with something for a while, but we haven't, give us a hand. Help us get up to speed so that we can work with you. That's our intent. But we are skeptics as well. And so we want to be convinced that you are indeed going to be delivering reliable, robust data that we can count on. And so expect those questions, but also expect the collaborative approach that we intend to be bringing to work with you to find out the answers.
Thank you, Owen. So Tarek, building off of the idea of bringing all parties up to speed, why is borrowing data beneficial? And specifically, under the current methodology, any insights that you have around applications that are most likely to benefit from the data aggregating model?
Yeah, so I'll start out with, I'm trained as a biostatistician, so I think there's been a lot of discussion about, in the past of what is the best data, right? Oh, you've got engineering models, that's the best data, and then you go to a clinical statistician and they say a clinical trial, a randomized clinical trial, that's the best data, and then you go to post-market studies and some people say, well, this is the best data because this is data in the wild. And so the question of which data is most appropriate, I think, is what we have to move away from and say every source of information you can get has some positives and has some negatives. And so truly when you want to get to the best quality information, it's combining data and figuring out a way of appropriately putting all of that data and all that information together. That's what our brains do, and that's what we should do as developing evidence. And so when you do that appropriately, you can get a lot of benefits. You can get to getting innovative devices to patients earlier. And you can also see how you can get a better understanding of when you do get a successful clinical trial, if that's going to work well in the general population or in the wild. I think if we really work on these methods, we can really help move the needle with some of the work that we saw from Jeff Shuren at the beginning on how we can sort of bring back that pre-market trial and integrate things together, because this approach would allow us to seamlessly go from modeling to pre-market to post-market and then iterative products beyond that, and keep that cycle just moving. But you've got to be very careful about doing it, and we have to be prudent about how we combine data. So that's really what the MDIC working group has really been about, is you've got to really first start with first principles. Is the information we're using to leverage to augment the clinical trial, is it exchangeable? And not necessarily in a statistics way, but in just a first principles. These patients have similarities. We're using the product similarly, and it might be slightly different, but it's got a similar feature and it's localized in the same place. Then once you do that, then within the clinical trial, you put some of these methods in place to protect against making mistakes, right? We think that product is the same, but as we run the trial and sequentially look at the two bits of information, we can see that there might be discrepancies. So now we need to start downweighting some of our assumptions that we thought. And so if you can do that in a really well-designed study, you can really get the benefits of all the sources of information. And that's really what it comes down to, is what types of endpoints is really, and what type of data can you borrow? It's really about its specific application, but you really need to have exchangeability and at least some sort of idea of how those can be exchanged.
Thank you, Tarek. I skipped a slide. So, Michelle, kind of going off of where Tarek was going and what Don was talking about earlier, as one of the founders of the computer modeling group within MDIC, how does the idea of this virtual patient model address ethical considerations impacting patients and facilitate real-world evidence development?
Well, first of all, I just have to say it's so exciting to see the fruits of all of this work come together the way it is. MDIC has just gone so incredibly far in such a short period of time. I'm seeing all the smiling faces from Bill and the board members up front. But this is one of the areas that I think was, from a regulatory science point of view, one of the most challenging MDIC took on. We had this vision that if you could develop a virtual patient, a patient that was able to mimic some of the physiological conditions, some of the pathology of the diseases you were trying to face, and use that in place of actual patients in clinical trials, wouldn't that be an amazing feat? And I'm looking over at Kyle Myers, who was one of the founders within FDA of really thinking about this concept and hammering it out. And if you think about what Jeff presented, Dr. Shuren presented this morning, sorry, I know him so well, I just say Jeff. What Dr. Shuren presented this morning, moving that line of when you introduce a device is very, very important to the patient waiting on that device. But it's also important in other ways. It means that when you're doing that clinical trial, if you're able to reduce the number of patients you enroll by 16%, 25%, 50%, you're not just saving money and time from a company point of view, but you're saving exposure for those potential patients from a clinical trial. And clinical trials are not without risk. Now, from a benefit-risk point of view, up till now, we've considered that risk acceptable because we need the data, we need the information, and we all agree that a double-blind controlled study is the optimal way to get good data, speaking about the relative value of data. But to that individual patient who doesn't know if they are getting a sham device or the actual therapeutic device, if they are getting a real drug or they're getting a placebo, that can have really severe consequences for the course of the illness in that individual patient, not to mention the time that they invest in the trial and using their limited health and financial resources to participate. It's a huge thing to ask of patients to participate in a clinical trial, and I think too often we gloss over that because we know how critically important it is to have confidence in the devices and medical products we're introducing into the ecosystem. But if we can find a way to reduce the number of patients that are exposed to that risk, it is a very, very valuable thing to do for everyone involved. And so imagine you're saving patients from being exposed to the risk of a clinical trial. Then you're also introducing the device sooner, because the more patients you have to enroll, the longer a trial is going to take. And that means you're getting it to the waiting patient population sooner, which is a huge ethical bonus. And in addition, you're getting that real-world evidence, that data in the wild, faster, because you're able to see how that device or that medical product really operates in the complete healthcare system with providers who may be very, very skilled, like most of our clinical trial sites and academic medical centers around the country, as well as, the more remote physician who might not get access to the training and information to use those devices and products as expertly as some others the very first times they use them. So what we really want is to shift that whole paradigm earlier in the process to, 1, reduce the number of patients exposed to clinical trials, 2, reduce the waiting time for the patients who really need those interventions, and 3, get access to the real information we want, the information on how that device really performs sooner rather than later. And from an industry point of view, coming from J&J, we want to know as soon as possible if one of our devices is not performing the way we expect it to. We want to have that information, we want to act on that information, but we can only do that if we speed the entire cycle. So that is, looking at it from a patient point of view, that's a huge set of benefits that we should not ignore. And then if you think of it from a societal point of view, decreasing the cost of introducing these new medical devices and inventions, decreasing the time that it takes, all of that is going to help the healthcare system better afford to care for a larger range of patients. So this is incredibly valuable work. I really applaud Don. The reason I clapped when Don Bardot came up was because Don had to believe in this when no one else did and keep going when it was really a hard struggle. But the results speak for themselves, and it's fabulous to think that we'll get to a point where we can model a patient and save a real patient.
Thank you so much, Michelle. And Lisa, as the first organization to successfully complete a retrospective analysis using MDIC's virtual patient model, can you discuss the characteristics needed to create buy-in and position an organization for success when undertaking study design innovation?
Sure, thank you. And I'll be speaking from the diagnostic standpoint, and I think sometimes it's easy to think about with implantable devices, the complexity of the trials that need to be done with implantable devices.
Closer.
Pardon me?
Mic a little closer.
Okay. It's got a knot in it. We easily can kind of grasp all of these challenges and issues with the growing evidence requirements, the cost of doing studies, et cetera. In the diagnostic arena, things have changed rapidly for kind of our solutions also. And while we don't have the long clinical trials as an implantable device has, what we do have, and the challenges with evidence requirements, is the enormous number of assays that we develop, the broad spectrum, the different patient populations and target organisms that we have to go after do require patient enrollment into our programs and into our studies to be able to collect the samples, to do all the sensitivity, specificity analyses, et cetera. And the growing evidence requirements are applying great pressures for us also in the diagnostic space. So some of these ideas around the virtual patient model and modeling how you translate that into the diagnostic space have been very exciting for us. And our involvement, Anita Besselmeyer and Val Parvu and some others from our team that have worked with MDIC and the folks that I'm sitting with to look at some of these models have made some very interesting progression for our space also. And we did do a first pilot of a retrospective analysis in this assay space where we said every assay or next generation assay that we do, typically we're running the same size clinical trial. And again, the volume of studies for these assays is enormous. So we looked at retrospectively at a next generation assay and said for the original assay, if we we had borrowed data or information in the sensitivity arena from that original study, applied it through Bayesian adaptive trial design, to this next generation assay, would we have been able to increase our savings around reduction in the number of prospective patients we needed to enroll, et cetera. And we did complete that analysis. The team did demonstrate, as I think it's showing up here, is that we would have been able to stop the trial at half the number of patients that were required to meet the sensitivity endpoint. And by reducing that sample size in half, we would have shortened the enrollment time from 29 weeks to 16 weeks. And you can easily start to translate a savings of just the little over 3 months enrollment time into shorter time to database closure, the cost of keeping study sites open, et cetera. So there are great opportunities in applying these new methods. So it was a wonderful demonstration for us to take within and across the corporation to start teaching not only our clinical research staff and regulatory partners about how you think about when you apply these new methods, because it's not applicable to every situation, but where are there great opportunities to start applying these new methods? And then where does it make sense from a regulatory pathway standpoint with all the progress and partnership with FDA in accepting new methods? How do we start to move down that pathway? And while to be able to implement it and kind of run, if you will, not with scissors, but very carefully, how do we start running with real examples? There are a lot of operational changes that we would implement. How do you bring in the skill sets and the talent across the organization that understand these new methods, can apply them, et cetera? How do you change some of your infrastructure around procedures and processes, and how you stimulate and trigger thoughts about how do you use new data? Where do you go down a new regulatory pathway? But really what's imperative in kind of changing the paradigm and the acceptance from within a corporation is how do you translate and educate on these new methods and ultimately kind of answer the question, how do I interpret the results of these new methods to various stakeholders beyond the scientific minds, the statisticians, the methodologists? And there are probably two key stakeholders. One is the business leadership. It's ultimately a business decision on how I invest in taking a new pathway. Do I take a risk of applying some new methods and maybe being the first or even the second to go down that regulatory pathway? And what's my probability of success? So how do you educate and train at a business leadership level on how we apply this? And these types of demonstration projects with real data has been very successful for us, and we're looking at doing similar retrospective analyses to be able to share those results throughout. And then the other stakeholder group that is important to the business leader are the providers and those other customers that ultimately need to understand the new methods and know how to interpret the results, and that will drive appropriate adoption and use of our new technologies, whether those be assays or implantable devices, et cetera. And we do have some work and effort to be able to make that translation to the providers, to our customers, to the scientific community, so that we're confident they understand it and they will accept the new methods. I loved Roseanne's path that way and talking about paint and shades of paint, et cetera. Those are types of translations in different languages that we're going to have to think about. How do we go down that path also to convince our customers for all the right reasons and have them feel confident to be able to use the products?
Thank you so much, Lisa. And thanks to the entire panel for their insights. If we have time for it, I would like to open questions to the floor for any or all of the panelists. Bill?
I can just speak loud.
Okay.
It won't come over the video. sometimes I think that because we talk about the reduction in the number of subjects, it's inferred that there's more patient risk. But on the other side, I believe that one of the benefits of this approach is that there's additional knowledge and insight before you start the clinical trial. So in fact, the actual understanding of the performance prior to starting the clinical trial is potentially higher than it would be for the traditional approach. Am I interpreting that correctly?
Yeah, so that's exactly right. If you think about two sequential trials, right? You've got your historical data, and you run this trial and you say, we're going to borrow some information. And because of the loss function, you're only allowed to borrow data if you can prove exchangeability along the way. So at the end of the trial, you have success, but not only do you have success in the trial, but you also have two sources of information that show that you have similarities. And so that actually, in the end, gives you more information than you had at the beginning. So very powerful.
Hi, I'm Bram Zuckerman from FDA. I direct the cardiovascular division. I really enjoyed this panel because while the promise of Bayesian adaptive approaches is certainly evident in the fine presentations of Dawn and Roseanne, we heard about some practical challenges, which is the main one being predictability of FDA to this new approach. So I would like to underline several points because this issue came up yesterday in the Chief Medical Officer's meeting. Number one, conceptually, the use of the power prior, as noted, has been a real advance in Bayesian statistics for a variety of reasons. However, it is a new type of procedure such that what I would recommend is if your corporate leaders still have reservations, there are several possibilities. Number one, it's vital, as Owen mentioned, to use the pre-submission process. Number two, our new chief of biostatistics, Dr. Ram Tiwari, is an expert in this area. In fact, his publications over the last 5 years have basically been in this, in the use of this methodology. Number three, certainly I would recommend you also ask for him to be at the meeting if you'd like. Division directors can attend. And another option that Dr. Carlson brought up yesterday is certainly we can run this methodology prospectively in parallel with standard frequentist methodology to see what some of the potential practical roadblocks are. So I think the basic theme is that MDIC has made a very important contribution here, getting all stakeholders to consider a new way of thinking here, but we need to continue to work together. Thank you.
Thank you so much, Bram. Other questions?
This question is to Roseanne, but one of the projects showed a 16% reduction in the cost of the trial in terms of sample size. Trials are needed not only for the regular submission, but also to payers, getting into the guidelines and so forth. At what level? And any saving is a good saving, but the flip point of this is it makes the data harder to interpret, particularly by the medical community at large. So is there any advice here on what level of saving would make it worthwhile for somebody to make the trial more complicated and the data more difficult to interpret? Don? All right.
So one of the things in my particular role when I design clinical trials, I'm not only looking at the regulatory approval, I'm also looking at the reimbursement and also making sure that we're convincing in terms of payers. And one of the things that actually CMS has been focused on, particularly in the last couple of years, is this type of data aggregation and using Bayesian in terms of looking at what is going on in the overall field in terms of data. So when you say it's not less evidence, it's actually the same amount of evidence with the same level of credibility in terms of its interpretation, and also in the interpretation of our particular payers, where we're talking about private and public. In fact, they are encouraging this because it allows them to not look at just individual trials that say different things, but how do these individual trials contribute to each other? And they have greater confidence. I was in a panel in which they were talking about how many more Bayesian statisticians they needed to bring in to CMS and how important this was in doing this data aggregation for CMS to understand truly what all these individual trials mean in aggregate. Dawn, did you have something else you wanted to say?
So just to add to that, Naveed, as a MDIC board member, I'm going to throw a challenge back at you, which is in the first iteration of MDIC, it's been necessarily focused on the US FDA, but getting global regulators to accept this type of data is going to be the real pivot point. Because we're facing in China, for example, many of those folks around the room were together in China last week for the CIMDR meeting with CFDA. They are trying to really drive companies towards repeating clinical trials with the Chinese patient population. So now you're talking about really increasing the risk of exposure to clinical trials, not to mention the cost. And the moment you're able to say there's a group of regulators around the globe who are willing to accept this data in lieu of their own type of trial or a trial with their own population, then you're really going to be able to make that case to business leaders. So maybe MDIC wants to lead an opportunity to spread the gospel to a wider range of regulators.
And just to add to that point, just for a second, also that at the beginning everything is new, right? So the idea of how do we educate the general, group about this new method. And if you think about a lot of type of statistical methods, at the beginning they're like, what does this mean? But after time, they become standard. So you can, at the beginning, there probably will be some adoption issues and understanding how to market the product with this new method. But as you go along, those savings will continue and the methodology will be better understood to everybody.
This is Chip Hance. I'm the board sponsor for this particular project. I just wanted to say I think this business question is a significant one. We've done 4 retrospective analyses. There are several more that are in process, and that'll sort of quantify the value of what this can be and what some of the reasons why some studies achieve a 16% and some studies achieve a 50%. And we're going to have some, guidelines and rules of, okay, here's when it's best to apply. I think the other big issue that comes out that we need to take this step forward is, okay, how is the data actually going to appear in a package insert, and how interpretable is it by the general community? And I think if we can do some of these retrospective analysis and take it another step forward, and even have some discussions with FDA. Okay, so how would this actually appear? Because I think one of the criticisms from a business perspective about Bayesian stats is it looks like a lot of hand waving when you look at it in the end, and someone has this interpretation that maybe the sponsor was trying to hide something by using Bayesian statistics. And so I think it's really important that we take this one more step forward and make sure that we've got a legitimate way to present this in a clinical fashion. And then lastly, I'd like to get on the soapbox and say, I think everyone who's been a part of the retrospective analysis has gotten an awful lot out of that experience. And so I would encourage anyone else who is interested or intrigued by this conversation here today to approach Dawn or Roseanne or Dan and say, I might be interested in doing this in our company. And more importantly, we need people who are willing to do prospective forward-looking studies and say, okay, we're going to actually make this part of our IDE submission, we're going to go through the process, and we're going to actively consider doing this. So I think everyone here in this room, especially on the industry side, should actively consider this. Thanks.
Thank you, Chip. I think we have time for one more question, comment. Looks like Bram wanted to add.
Again, I think Chip's comments are quite good and very important, and the notions of predictability, transparency, understanding, and making the business and value case certainly can't be forgotten. But I would put it the other way around. I think there actually are many recent examples where this methodology has worked. For example, the one that I'd like this audience to look at as a homework assignment when they go home is the Watchman Boston Scientific left atrial occluder label. From a cardiovascular device perspective, this is a very innovative, transformative technology. The study was done via Bayesian technique. The problems that the trial ran into had nothing really to do with the Bayesian technique as opposed to trial execution. But I think if you look at the writings of our former stats director, Greg Campbell, you'll see that there are now loads of Bayesian examples. The Watchman example is another good example because many times at FDA we have one pivotal trial being done and we get indeterminate results. The Bayesian methodology through hierarchical modeling or use of the power prior allows for building upon that indeterminate result in a easier fashion many times than frequentists. So there's a lot to chew over, and I'm sure that there will be many discussions on this technique in the future. Thank you.
Thank you. So I think that's it for the panel. Let's get a round of applause for our panelists as well as for Don and Roseanne.